So we have our first official talk. We are going to start today with recently reported updates from Italfarmaco on Givinostat trial for those with Becker. Joining us to share this new data is Dr. Paolo Batica. If you have questions, you can submit them in the box below your player. For now, I'm going to hand it over to Paolo to start the presentation.
So Ryan, thanks a lot for the invitation, and thanks to PPMD for giving us the opportunity to share this top-line result from our proof of concept study with Givinostat in Becker muscular dystrophy. If you can go to the next one, please. This is my disclaimer that you have seen already. I'm a full-time employee of Italfarmaco, which is the manufacturer of Givinostat. Givinostat is currently in development for the treatment of Duchenne and Becker. And as a result of that, it is not approved for sales in any country around the world, including the US. And this presentation is intended for dissemination and discussion of scientific information only. If you go to the next one, this is a summary of my presentation today. So I will start by reminding all of us the results we have already obtained in Duchenne with Givinostat. Then give you an overview of our phase 2 study in Becker muscular dystrophy, move on to the top line results, and then conclude and tell you about the next steps. If you can go to the next one. Again, so I'll start with the summary of the results with Givinostat in Duchenne. Next, please. So Givinostat has been shown to counteract the muscle deterioration in Duchenne muscular dystrophy. We were actually able to demonstrate that at the histology level, so in muscle biopsies. This was true both in the MDX mouse, but also, and most importantly, in the Duchenne boys that actually took part in our phase 2 study. And after 1 year of treatment of Givinostat, on top of steroids, we were able to show that there was reduced fibrosis, reduced fat infiltration, increased muscle fibers diameter, and increased muscle regeneration. The kids then continued the study and are still on treatment now in their 8th year of treatment. And so we were able to confirm the result, observe the histology with MRI, and also to show that the long-term treatment with Givinostat is associated with a delay in loss of ambulation and a slower decline in the respiratory function. So given the similarities between Duchenne and Becker, and building on the experience that I just summarized, we decided to actually move on into a phase 2 proof of concept study to evaluate the effect of Givinostat in ambulant Becker subjects aged 18 to 65 and who were able to walk between 200 and 450 meters at the 6-minute walk test at baseline. If you go to the next one. Again, next one again. This is a description of the study. The objective of the study was to demonstrate that Givinostat can preserve the muscle and slow down the disease progression. And this was actually assessed by means of histology, muscle biopsies done at the beginning and at the end of the study. MRI, also this done at the beginning and at the end of the study, and then a number of functional tests that were done every 3 months, and you can see the list on the right-hand side. So after a period of screening, the patient that actually met the inclusion and exclusion criteria were randomized in a 2-to-1 ratio to Givinostat or placebo. This was actually given as an oral suspension, and then the follow-up lasted for 12 months. If you go to the next one. This is the disposition of the patient in the phase 2 study. We screened 70 patients, to then have 51 that were randomized to either placebo or Givinostat. And as you can see in the slides, the 17 patients that actually were on placebo completed the study. And in the givinostat arm, 30 patients completed the study. The reason for withdrawal are at the bottom of the slide. You can see 2 patients were actually withdrawn due to an adverse event. These were in one patient platelet decreased, and in the other one triglyceride increase, which is a signal that we have seen previously. And when the patient stopped the drug, the adverse event resolved completely. The 2 other patients actually had to withdraw from the study because they were, in essence, they had to travel from one country to another to complete the study. And obviously, because of COVID, all the traveling were not possible. And so we had to withdraw the patient from the study. Next slide, please. This is a description of the baseline data of this patient. As you can see on the left-hand side, the age ranged between 19 and 61 years of age. On average, the patients on givinostat were younger than the one on placebo. If you go to the right-hand side, you see the baseline value in the different parameters that we measure. On average, we have approximately 25% of fibrosis in the muscle biopsy of the upper arm. The MRI fat fraction of the whole thigh, the fat fraction was close to 60% on average, so a highly infiltrated muscle. And then if you look at the function test, I guess the important information is to see that there was actually a high variability. So then if we look at the 2 groups and make some consideration about the baseline value, we actually had a significant increase in between the groups, a significant imbalance between the groups in histology. So the randomization on histology did not work as well as we wanted. We looked at whether there were correlation between the different parameters at baseline, and the histology, MRI, or function did not correlate with age or time from diagnosis. This is something, by the way, that has already been shown in Becker. And then we also looked at whether histology and MRI were correlated with function and strength, and the answer is yes, they were highly correlated. And also this is replicating data that have been previously shown by others. If you go to the next one, then we move to the result. Next again. So this is the primary endpoint based on the experience with Duchenne muscular dystrophy. With givinostat in Duchenne muscular dystrophy, we selected total fibrosis as a primary endpoint. But as you can see in the graph, on the left-hand side, firstly, you can see that actually the fibrosis was actually larger in the placebo group compared to givinostat. So the 2 groups were in balance, as I said. But I believe even most importantly, there was actually no change in the 12 months. So this parameter did really not change throughout the 12 months. And as a result, also the 2 groups were not different. But if we go to the next slide, we can look at the MRI results that are actually telling a different story. So if we look at the fat fraction in either the whole thigh or the quadriceps, well, first of all, the parameters were actually, the fat fraction was balanced between the groups at baseline. And also, if you look at the right-hand side of each graph, you can see that there was a significant increase in fat fraction, so in fat infiltration in the placebo group. And this actually did not occur in givinostat. And as a result of that, the 2 groups were actually highly significantly different. If you go to the next one, we can see the results in the contractile cross-sectional area. So the fraction, the portion of the MRI image that is occupied by muscle. And also here for both the whole thigh and the quadriceps, the results were balanced at baseline, and there was actually a deterioration in the placebo group, which was not observed in the givinostat group. And this was highly significant in the whole thigh and was nearly significant for the quadriceps. So if you go to the next one. As I mentioned at the beginning, we did a number of tests, which are the standard tests being conducted in particular from the experience in Duchenne. Also here, we have the same situation as for histology. So there was actually no change over time in this test, the 6-minute walk distance, the Forster climb, the time 10-meter walk run, and the time to rise in either givinostat or the placebo group. This, by the way, was also true for the strength, where no change over time was observed. And also here, at the end, we did not have a difference between the two groups. If you go to the next one, the situation was actually different with the standing and transfer domain in the motor function measure, which is a 32-question questionnaire that is looking at different domains related to mobility. And as you can see here, in this case, we did see a decline throughout the time of this parameter. And if we look at the result at month 12, the difference between the placebo and the givinostat group, the decline in this group was actually slower than in the placebo group. If you go to the next one, we can then look at the safety. In the 51 patients treated in the study, we did not have any serious adverse event or death. The most frequent adverse events are the one that we actually I have shown other times in Duchenne. So diarrhea, platelet decreased, and increased triglyceride. We actually reduced the dose from approximately 70 milligram twice a daily, which was the original dose, to 50 milligram twice a daily to reduce the incidence of the adverse event. And we obviously measure a number of other safety parameters, including ECG, and we did not see any change in those parameters in the givinostat group. So if you go to the next one, again, please. So overall, we can conclude that the study clearly failed the primary endpoint, but we believe that actually this was due to the high variability of the histology parameter with a significant imbalance between the groups at baseline, and also by the fact that there was actually no change over time in this parameter. As I said, the picture is totally different with MRI. If we look at the MRI of the whole thigh and of the quadricep, we can confirm the ability of givinostat to counteract the muscle deterioration, also in Becker, by preventing fat infiltration and muscle atrophy. When we looked at the function and strength, we saw that actually there was no impairment or a very slow impairment, if any. And so this made it very difficult, if not impossible, to demonstrate a difference between the two groups. The picture was different for the standing and transfer domain in the motor function measure, where the results are actually suggestive of a potential benefit of givinostat. The potential benefit is also suggested by the fact that the MRI is actually highly correlated with function and strength at baseline. So we believe that the results that we obtain in MRI will eventually translate also into a functional benefit. And as I said at the beginning, the givinostat treatment also in Becker individuals continue to show a good safety profile. So if you go to the next one, these are really the next steps and also the relevance, obviously, to the ongoing evaluation in Duchenne muscular dystrophy. With regards to Becker, Italfarmaco will, on one hand, continue to evaluate the data. We have many more analyses to conduct, and then also to discuss with the European and US regulatory authorities to define the best path forward for givinostat in Becker. And obviously, with regards to the Duchenne, this was clearly a standalone study, but I believe that the results are actually confirming the beneficial effect of givinostat, and this, if you want, replicated what we had observed already in Duchenne boys. And with that, let me, if you go to the next one, let me thank, obviously, all the patients that are enrolled and have conducted our studies. Their participation has been, obviously, key to the development of this drug. Clinical trials sites have done an excellent job, in particular if we consider the pandemic situation, and obviously a big thank to all the patient organizations that have actually supported us. Let me also remind you that this study was funded together by Italfarmaco and by the Regione Lombardia with a grant coming from the European region. And at the bottom, you can see the place where you can find more answer if you want to know more about the studies or any question you might have. And with that, I'm happy to take any question. Thank you.
Thank you, Paolo. This is certainly a challenging population to work with where we don't have robust natural history data. And as we saw in your presentation, looking at something like total fibrosis over 12 months, there's not much change, but we did see some of that with fat fraction. And so kind of two questions for you. One is, as you think about moving forward, is something like fat fraction a preferred outcome measure to try and use? I mean, obviously, we do have to still work on it being qualified. And the other question is, we saw, again, 12 months, hard to see change. Is this a population where we have to think about longer trials, which can be challenging for families and for those patients? But would love to get your thoughts on that.
Eric, you have touched all the different questions which are running in my brain since I see the results. So I think you got them perfect. So let me start from MRI. I think MRI, these results are confirming the value of quantitative MRI. I think they really are a very important tool in our muscular dystrophies to look at the evolution of the disease in the muscle, which is a key tissue that is affected by these diseases. And I'm actually a strong supporter of MRI as a potential endpoint also for the future. And I think this applies to Duchenne, but also applies to Becker. With regards to the evolution, I think that the point, you said it quite clearly and quite rightly. We have limited history, natural history in Becker. And what we saw in these patients, so these patients were selected as patients that were, in essence, doing still fairly well on a 6-minute walk test, because they were walking between 200 and 450, which is not a bad value. But for example, if you look at the MRI fat fraction, they had 60% of fat infiltration. So there is probably these patients, their muscle are severely affected, but possibly they have better compensatory mechanism. And so we will have to think about how to best assess the effect of a drug in this population. And this is what we will have to do in the future and also discuss with the regulatory authorities.
Great, thank you. I think we'd also just like to hear your experiences. We're seeing some more companies as well look at Becker populations. In terms of recruitment, was that a challenge with this population, or was it something that was easy to fill in your enrollment?
Well, I think I have to answer to the question. The first one is that the number of patients that are willing to take part in the study is large. because as you just said, there is actually very limited clinical studies ongoing in Becker. Obviously, the issue comes when you start selecting what you believe is a more responsive population. As you have seen in our trial, we have, in essence, screened 70 patients to select 51. And by the way, there was probably an even larger group that was pre-screened. But I mean, given what we just saw and this very slow progression, I think we will have to identify which is the best way to recruit patients that actually can show some effect in a decent time. I mean, I don't think that anybody of us wants to run a 3 or 4 years placebo-controlled study. I mean, that would not be possible.
Yeah, no, I agree. Paolo, have those patients stayed on drug and open label? Any of them drop out?
This is actually not, we did not start an open label because, as I said, this was limited to the funding received from the Regione Lombardia. But obviously, this is something that we need to reason about for the future. Yes.
Sure. And there's just a follow-up question. I don't think that many U.S. patients are familiar with the motor function measure, or MFM, that's been used here. I was wondering about taking this forward with regulators. Have you had talks with regulators about that measure, and any comments in regards?
So this is actually a scale that is used in Europe. I believe the originator are from France. And actually, the EMA was keen for us to have it also in Duchenne. You are right, it's not that familiar in the US, and this will be a discussion for the future with the FDA. But I guess to me, the important point is that it looks like another type of questionnaire, which, let's say, looks more at, in a holistic way at the changes in the disease, might be better suited in Becker. I think it still needs to be confirmed, but these original results might suggest that.
Great. Well, we are grateful that you're studying this population, and we are here to support however we can. So thank you so much.
Don't worry, I'll come and knock at your door.
Yes, and thank you for helping us with the guidance. We know you're going to be involved in that process along with many other companies, so thank you.
Thank you.
Okay.
Thanks. Thank you. Okay, thank you.
Great.